You can start, madam.
Yes, yes, yes. So I'm just sharing the screen right now. And so our purpose is, let me start from beginning. So a warm welcome to all of you to this class of advanced bioinformatics. So before going through the deeper roots of bioinformatics and advanced bioinformatics, we should understand that what is bioinformatics before going into advanced bioinformatics, right? So I will just today give a brief introduction of what are the fields of bioinformatics, what is an advanced bioinformatics, and what are the softwares, tools which we will be learning, utilizing in our projects, in our everyday life when we will apply bioinformatics knowledge and skills. So what is bioinformatics? So basically bioinformatics is a combination of basically two major fields, biology and information technology. Okay, so whatever we understand and learn in biology with the help of information technology, though it encompasses computer science, statistics, mathematics, etc. So what do we learn? The first thing is, what do we learn in biology? So everything, genes, proteins, molecules, all the biological data. So whatever data is within the bioinformatics, whatever data is within the biological science, that is majorly understood by information technology. Information computers are able to analyze and interpret that biological data. So that is called bioinformatics. And why this field came into existence? So earlier what happened, that when human genome project started, so there were a vast amount of data, and it could not be presented, collected, analyzed, etc., and distributed on the papers. So this bioinformatics field came into existence. So nowadays, this bioinformatics has many sister fields, and it has grown a very large scale. And there are many high throughput sequencing technologies which are utilized within this very field. So actually, you will ask that, okay, ma'am, what is this high throughput sequencing technology? What do we mean by this? So any technology which utilizes sequencing, and again, the means we have many sequences, right? We have a whole, suppose human body, any organism, it has the whole body has many sequences. There are many genes, proteins, there are many small molecules, and that all has to be analyzed. So sequencing comes into existence. So the technologies which are used to put all that knowledge into existence, they are known as high throughput sequencing technologies. So I will just let you know that what are the key areas of bioinformatics, first of all. Okay. So so everybody knows that what is bioinformatics, what everybody knows this definition. But in our syllabus, we are going to learn, since today is the beginning day. So I'm just going to give a brief introduction to the key areas, software tools, etc. So what are the key areas of bioinformatics? So as I told, that high throughput sequencing technologies, we should focus at this term. So sequence analysis, if we are talking about. So this is an important area which is considered inside bioinformatics. Why? Because sequencing is something which is very much needed by by scientific world, and everything like proteins. Our body is made up of building proteins are the building blocks of our body, right? Or any organism. So if we are going to understand that, okay, what is the nature of that organism, what is the whole structure, what is this a small, even from the single input to a larger output, how we are able to analyze that. So it begins from the proteins, or it begins from the DNA or genes. So in that case, sequence comes. So what is sequence basically? So sequence is a small stretch of a DNA, of gene, of proteins, amino acids, etc. So that sequence needs to be analyzed. So in order to analyze that sequence, today many technologies have arisen, like genome sequencing and assembly, and this NGS sequencing, metabolomics, many things are analyzed. So we will understand that how we can analyze, we can understand that uh, means assembly of that, how we can understand that sequence, how we can analyze it. So one of the methods is genome sequencing. So what is genome first of all? So genome is the whole set of DNA, whole set of DNA, total DNA within an organism's body. So
Hello, ma'am.
suppose,
Ma'am, your slide is not moving. Your slides are not moving.
okay, it is not moving.
No.
Okay, I'll, is it visible right now? Hello?
First slide is there, I think. Bioinformatics is an interdisciplinary field that combines, and
Yes, sir.
that ad is also looking there. Ad, I think ad is there because that sentences are hide behind the ad.
Okay, let me go beyond it.
Because combines after that bio and second line statistic too.
Right, right. So I was
Uh,
telling about, I was actually said I had few tools, means because right now I am able to present a single slide PPT. So I was planning to present few, since it is Microsoft Teams. So I was planning to present something editable tools also. So it was, the planning was within Zoom. So that's why this is the limitation that I'm telling through PPT only. So that's why I'm focusing on it.
okay, ma'am, continue.
Okay, so here we come, genome sequencing and assembly. So so as I told that, what is genome? Genome is the whole, the total amount of DNA within within the body of an organism. So how we sequence it, how that whole amount of DNA is sequenced, and how it is assembled, that comes under genome sequencing assembly. So it is assembling short DNA sequences into longer genomes. So sequence analysis, the first point comes genome sequencing and assembly. So you will ask that, okay, what is the purpose of assembling these short DNA sequences into longer genomes? What is the purpose? So of course, the purpose is that we need to analyze that data. We need to analyze that data in
Hello,
order to
hello, madam.
interpret that.
Hello,
Hello?
hello, ma'am.
Yes. Yes, sir.
Scroll down that slide. If you are directly online showing, scroll that slide so that because see here that slide bar is here to move the slide or like that. Okay, so in this way. Yes, now continue.
Okay, so the second field is structural bioinformatics. So this is also an important field. So you must have heard about that, about many softwares and tools like Chimera, like which AlphaFold 3D AlphaFold of this right these days, Google had. So if we have unknown protein structure and we predict the three-dimensional structure of proteins, or then we have software for that. So that comes under structural bioinformatics. So under structural bioinformatics, we can predict the protein structure, and we can also model the molecules. So we will gradually come the software, we will gradually come through the process of understanding those software tools which are understood through protein structure prediction and molecular modeling. Then comes functional genomics. So in functional genomics, there is a field known as gene expression analysis. So in this, all the biotechnology students must be knowing that RNA sequence microarrays are built, and that is done in order to measure and analyze gene expression levels. Okay. So then comes gene ontology and pathway analysis. So according to this, genes and understanding the biological pathways are involved within. So that is done in order to understand the function of the genes. So that is the whole pathway. So there are many pathways, the whole disease, to which pathway which disease will be linked. So that is built under this gene pathway analysis. Then, so there is comparative genomics, right? So under comparative genomics, there is evolutionary studies. So suppose we understand the nature of the genome of one species, and we want to compare it with the other species. So in order to study the evolutionary relationships and to compare the history of both the two species, we use these evolutionary studies, and it comes under comparative genomics. So this is also one of the fields under which is studied under advanced bioinformatics. So then it is conservation analysis. So under conservation analysis, conserved sequences may have importance. So what are conserved sequences? Means the sequences which are not changed. Up to, in certain organisms, the sequences are not changed, and they prevail up to generation. So that is studied under that is analyzed, and those sequences are preserved and they are analyzed. They are they are made as templates, and that is done under conservation analysis. Then comes systems biology. So systems biology is very much important concept within bioinformatics. So like network analysis. So we study biological networks, like what are protein interaction networks, what are metabolic networks. So there are many software tools like cytoscape, string databases. So according to that, we study network analysis. So we form the networks between different proteins, and which protein links to which disease, which protein links to which disease. That whole network, that software analyzes for us.
Hello, ma'am.
Hello.
Hello.
Yes, sir.
Hello.
Hello.
Next slide.
Then there is data management and integration. So database development, data mining, and machine learning, these are also parts of.
Hello, ma'am. Hello, ma'am.
Yes, sir. Yes, sir.
Is that on slide, data management? Because that slide is not looking here on the screen.
Okay, it is not looking. Let me. Is it visible, sir?
That structural bioinformatics, protein structure prediction, that is there on the screen.
Okay, I think there is network lag. Is it visible right now?
No.
Okay. Let me stop sharing and reshare that slide. Hello. Share. Hello, am I audible?
Yes, you are. Yes, you are audible.
Okay.
Just try to move slides, ma'am. Then you start.
Okay, yes.
Take next slide.
Let me some. issue with the slides, I think. Yes. Okay, sir. Let me directly jump into the syllabus.
Do one thing, ma'am, today.
Okay, yes, sir.
Yes, is it possible to share?
Yes, sir. I can share the syllabus. Actually, the slides which I had prepared that are not coming. They are not moving only due to some network lag, I think. So I had just give me 2 minutes. So the screen is under sharing.
Screen is
So
visible,
this
but you are
is,
shared here that computer screen, not presentation. Share presentation screen.
yep, the presentation is not coming only, sir. Uh, that application has closed. I'm trying to reshare it, but means some restrictions are there, I guess.
So that
So
previous slide, previous PPT, same PPT.
yes, same PPT, sir. Same PPT. I can, although I had made
Okay, okay,
those notes, so I can
okay.
just try to explain from here if I. But that was in detail. That was much detailed portion which I was covering for the introduction to advanced bioinformatics. Okay, so I was talking about this emerging trends and role of software tools in the bioinformatics. Uh, Let me try to bring that. So that PPT, I lost that PPT actually. It's, I'm so sorry. Uh, can I
Hello, ma'am.
share on
Hello,
the group?
hello, hello, ma'am.
Yes,
Hello, ma'am.
yes, sir.
Just give about introduction about you, brief introduction, and what content you are going to teach in this course.
Mhm.
Today,
So
just discuss that only what you are going to teach in this 30 hours or 1 month course. Okay.
yes, so basically we are going to learn the advanced bioinformatics in which we are going to see the various
Hello, ma'am.
softwares and tools.
Hello.
Yes, yes, sir.
Ma'am, you can share that syllabus PDF for that.
Is it visible? Is the screen visible?
Yes, it is visible.
So this is the content portion in which we are going to see the introduction to advanced bioinformatics. So I will be telling about the various softwares and tools which are going to be which are upcoming in the fields of bioinformatics, like sequence analysis, then structure prediction, then molecular modeling softwares, then protein interaction databases and softwares. Then the role of those softwares in bioinformatics research we will be seeing by taking small examples. Then we will be seeing that what are the challenges that may come when we use such softwares and tools. Later on, we will be moving further to next generation sequencing data analysis. So according to that, we will understand that what is NGS data, what is next generation sequencing data, and what are the why we need to analyze data, and what are the machines, what are the softwares like Illumina and other softwares that are used to pre-process. And we will also see some softwares for quality control of NGS data. So that we will be seeing at least 1 to 2 softwares we will be seeing in this case. Then we will be seeing the alignment and mapping of sequence reads. Small how we will be seeing what are the how are these sequence reads have different variants and how we call those variants. And we will also be seeing that those structural variants, that which are the structural. So these are the 3 uh, software tools for NGS data. We will be seeing these understanding of these BWA, SAM tools, and GATK. Okay, and these are basically transcriptomic and analysis, NGS data analysis. These are basically the fields which come under advanced bioinformatics. These are not the simple bioinformatics, but advanced bioinformatics. Then transcriptomics, we will understand what is transcriptomics, and then we will go to our number of projects today. Number of research is coming under RNA-seq data. So what is RNA-seq data and how it is processed within whole means this many projects are there under R for RNA-seq data. So we will first of all understand that how the whole those data within an organism is processed, and how we understand the gene expression analysis of that. So there are also softwares like DE, Sec2, and Salmon and Flare. We will also see a brief introduction of this. And these are few, we will understand the concepts, and then we will see the software tools along with that. Then we have epigenomics and chromatin analysis. We will understand the basic terminologies, and then we will see these omics data, and then we will see these softwares, Bismarck, Homer, and this Explorer, Hive Explorer. And then we will be coming to structural bioinformatics and protein structure prediction. So there are many softwares coming in this field. So we will. So what is structural bioinformatics? Small brief introduction I told today also, that what is and how the protein structure is predicted. If there is unknown protein structure, unknown sequence, and we have to predict its structure, then what are this? And under this comes the drug design approaches. What is structure-based drug discovery? What is uh, this and how it is analyzed, how they are how which are the software tools which are used for this structure based drug design. What is drug discovery? Homology modeling, ab initio modeling. These used to be means old techniques, but we will see the new techniques, like modeler is also one of them. Rosetta server is there. So these we will see, and later on we will see through small examples. Later on we can build up projects using these. Then we will see proteomics data analysis. What is proteome, and what are these basically these terminologies come under in biotechnology also. So what are mass spectrometry? What is the quantitative aspect of this proteome, that we will see. Then we will see few papers related to protein interaction analysis, and how protein to gene interaction is done. Then accordingly, we will see this as today I was telling about cytoscape. So there is also string database, these max quant, skyline, and others, but we will see cytoscape, one of the papers related to cytoscape. Then there comes network biology and pathway analysis. So here we will see the protein interaction networks. So there are many diseases which are many diseases are there, and there are many plant variants which cure those diseases. So if we construct such networks through which we analyze that which are the proteins which are directly linked to those plant variants, and they help they form a interaction with they act as curable to those diseases. Then that we will see under this construction and analysis of biological networks. Then the same that begins with the genes, so that we will see under gene regulatory networks. Then there is pathway enrichment analysis and functional annotation. So there are software tools. The name of the software is pathway enrichment. Uh, so that we will see under this. There are software tools under this. Yeah, Cytoscape is there, string database is there, what is Reactom. So these form the networks and pathway analysis that we will see under network biology. And this is also a very happening field today, and a lot of research is going in this very field. Then metagenomics and microbiome analysis. So there are subs like this software QUEEM 2 and Meta PHIAN and this. So these are very upcoming tools. So these are used to this very metagenomics is very happening field, and it is basically used to analyze the sequence related to microbiomes. It is very in Hindi we say it's sukshma. So very sukshma things, very not means we can not imagine also, and that is these software tools do to analyze those. So that is metagenomics and microbiome analysis. Under that, we will understand that what is the diversity between various microbiomes. So that we will see under this. Then comes machine learning and deep learning. So machine learning and deep learning is utilized everywhere. It is a big buzz. So we will see that how it can be utilized in bioinformatics. So basically, there are lots of research goes in cancer, in biomarkers. So that we will see TensorFlow, PyTorch. So little I will give a brief introduction of Python, and then we will come to PyTorch, TensorFlow, etc. So in order to, we will see a small example of biomarker discovery and prediction of protein structure and function using machine learning and deep learning techniques using TensorFlow and PyTorch. Then multiomics. Yes, so multiomics are basically the identification of molecular signatures. What are molecular signatures? There are various diseases, they have some molecular signatures. So genomics, transcriptomics, proteomics. What are these and how they come under the all these omics terms? How bioconductors are utilized to integrate these omics. So that is that, and analyze the whole data. So that we will understand, and we have software tools like bioconductor and omics integrator. We will see the introduction to these software tools. And then we have data interpretation and visualization. So we have ggplot2 and IGV and plotly. So we will see we will see a brief introduction of these three, and we'll see ggplot2 in details. So how they can be used to visualize interactively the biological data. So this is what the summary of the our syllabus is of this advanced bioinformatics, and I'm really sorry that I lost the content slides. I'll try to get it back and further continue. So sir, this is what is the summary of that, the whole syllabus, what I have given. So anybody wants to have any doubts or questions may raise their hands. Or okay, we have raised hands. So Amit Ladha, you can just unmute yourself and
Hello, good afternoon, ma'am. So I had three questions. Basically, first, will all the files be shared with us, which you have discussed today?
yes sir.
Secondly,
Yes.
will there be practice on the said bioinformatic tools that you have told us?
The tools which I first we will have an introduction, of course, and the tools which I mentioned, for that we will be I will explain you, and you will have to do the practice by yourself. And if you
Okay.
have some doubt, then we can, means of course we can raise it, and or you can ask on the
Ma'am,
group chat. Yes.
uh, so the third question is there any pre-reading that we need to do before, joining this course? Like any pre-reading books or titles that you can, suggest to us so that it can be faster for our learning?
That I will suggest you actually, along with your along with the classes, what are the pre-learnings that I will suggest on the group only. So that I will, of course, I'll mention that what should be the topics, or what should be the areas which should be covered. So any specific videos or books you can go through.
Okay, ma'am. That was all. Thank you.
Thank you. Yes, Barsha.
Good evening, ma'am.
Yes, good evening.
Ma'am, I was asking that the projects in which we will be given in we will be grouped to for a project. So, if I want to if I want to work on a research paper along with it, so can we get the guidance for that, or we
Yes,
need to
yes, you
do
will get the guidance. You will get the guidance at the last.
on our
We
own?
will do first. We will do that reviews. You will first. I will give you a structure according to which will have to prepare your research project, research articles, and accordingly you can go through. So we will decide the topics ultimately within the group. Within that will be done at the last of the when we will complete this syllabus. So, but I will giving you the hints that these are the key points where we can have our research projects. So that will be the thing. So you will be getting the guidance, of course. So no problem in that at all.
And that's all. That's all of my question. Until now.
Thank you. Yes, Amit, you have the question. Did you still your hand is raised? So I thought that.
Sorry, it was by mistake.
No issues. Uh, Anyone else have the question? Barsha, you can lower your hand. Rajat. Yes, Rajat.
Ma'am, will there be any doubt classes conducted after the classes? If suppose we have some doubts, so we can ask you in that afterwards.
What we can do is that every day, 40 minutes we can have class, and the next 20 minutes we can have for doubts. So will that be fine? For 20 to
Okay.
25 minutes, we can have doubts. So in that I will be, you can be asking your doubts, and I will try my best to give the answers or clues or hints to the you will have to again try for that, and keep asking me means continuously, so that your doubt ultimately comes, means you can solve it out, so that ultimately we will solve it out. That should be our purpose. Yes, Krishna
Okay.
Murthy.
Ma'am, hello, ma'am.
Yes, sir.
Hello,
Yes, Krishna. Yes, tell me.
ma'am. If for example, if in case like bio-edit tool, if you are teaching means, can you able to share that FASTA format file in individual mail or in this screenshot itself?
I will be sharing through in the form of docs. Either it will be Word doc, or it will be a PPT, or whatever will be suitable for the class, in which
Okay.
the images, in which the sequences I can post the means the complete answer to the what I taught in the class, that can be shared, and you can understand that. that in that format I will means apart from this teaching what we are reading in the advanced bioinformatics, not take it that means I will be sharing the recording will be go to you as well as the this what do we say the notes. The notes will be in appropriate format so that you can understand it.
Okay, thank you.
Yes, thank you. Yes, you can lower your hand, Krishna. Yes, Siddhi.
Yes, ma'am. I have doubt about do we have any reference book for this subject? Can you tell us?
There are many reference books. I'll give me 2 days time, 2 to 3 days. I'll post for on the group only, so you can refer it. Topic wise means appropriate videos, links, and books we have now that I will. So you can what you can do is you can have Kindle also, Kindle subscription. So in that also you can search for the books. So I will post it on the group, and you can all have Kindle subscription, and there you can just go through the books in details.
Okay, ma'am. Thank you.
Thank you.
Okay, tomorrow, ma'am, tomorrow's timing, ma'am will inform in the group. Okay, ma'am,
Yes, sir. Yes.
or same timing.
No, we can have the same time. I mean, 8 p.m. to 9 p.m.
Okay,
will be okay. I think.
okay, no problem. Okay, and today that whatever that PPT or that you can share in the group. Okay.
Yes, yes. I'm sorry for that. I'll just share that because basic I had, so I'll just prepare in the form of much understandable way, so that it is understood. It may be what doc. So I'll share on the this all only group only the notes. So
Okay.
maybe by tomorrow morning you will all get the notes. Since I would have shared right now only, but I lost that. So what I